Welcome, dear students, to another very important metabolic pathway, and that is the gluconeogenesis. The major source of energy in our body, that is the glucose. So, it is very important to maintain the normal blood glucose level in the body for the normal homeostatic functions. In case of fasting, if a person is fasting for just 2 to 3 hours and the glucose level in the blood starts decreasing, then in the liver of the human body, a process starts which is called as glycogenolysis, which converts the liver reserves of the glycogen into glucose, and then this glucose moves in the blood and it maintains the blood glucose level. But what happens when a person is fasting for a very prolonged duration of time, like 12, 20, 40 hours, or a person is starving, or he is having very intense exercise, or inadequate carbohydrates in the diet? Then, my dear students, even the glycogen reserves of the liver, they are exhausted, and in order to maintain the normal blood glucose level in the body, then the body starts converting some non carbohydrates into the glucose, and this synthesis of the glucose from non carbohydrates, that is called as the gluconeogenesis. This is not a process that is commonly taking place in the human body, but the gluconeogenesis takes place only when the glycogen reserves of the liver, they are exhausted, when a person is having prolonged fasting, starvation, very intensive exercise, or he is having inadequate carbs in the diet. So, my dear students, the conversion of non carbohydrates into the glucose, that is the gluconeogenesis. The word gluco refers to glucose, neo means new, genesis means synthesis or formation. So, the word gluconeogenesis means formation of the new glucose. It is a process in which the glucose is synthesized from some non carbohydrate molecules. My dear students, gluconeogenesis takes place in the liver cells, in the kidney, in the renal cortex cells, and in the intestines. Liver is a major contributor of the gluconeogenesis, and more than 50% gluconeogenesis in the human body takes place in the liver. But in case if a person is diabetic, then the kidney and intestines, they also become the site of the gluconeogenesis. Even in the normal persons, less than 50%, almost 30 to 40% gluconeogenesis takes place in the kidney and intestine cells. And within the cell, the gluconeogenesis starts from the mitochondria and completes in the cytosol of the cell. The first step of the gluconeogenesis always takes place in the mitochondria, and then it is completed in the cytosol. Now, the next point is that what are those non-carbohydrate molecules which are commonly converted in the glucose in the human body or in the bodies of some other animals. The first one is the lactate, but before the lactate is converted to glucose, the lactate is not directly converted to glucose. Lactate first is converted to pyruvate by an enzyme which is called as the lactate dehydrogenase. Similarly, glycerol is a very important substrate for the gluconeogenesis, but glycerol is converted to glycerol 3-phosphate, which is then converted to dihydroxyacetone phosphate, and this dihydroxyacetone phosphate, which is a triose, then it is converted into the glucose, which is an hexose. There are some glucogenic amino acids like alanine, glutamine. They are also converted to the glucose. And in human body, the 90% gluconeogenesis takes place with the lactate, glycerol, and glucogenic amino acids. And my dear students, some intermediates of the tricarboxylic acid cycle or Krebs cycle, they are also converted to the glucose. But all the intermediates of the TCA, they are first converted to oxaloacetate, and then oxaloacetate is converted to the glucose. In case of ruminants or grazing animals, propionate is the major non-carb which is converted to the glucose to maintain the blood glucose level. So these are some major substrates of the gluconeogenesis. But all these substrate, they are somewhat converted into the pyruvate before the pyruvate is converted to the glucose. So it means the first designated substrate of the gluconeogenesis, that is the pyruvate, or you can say the gluconeogenesis pathway actually starts from the pyruvate, and all other non-carbs by some metabolic processes, they are first converted to the pyruvate, and then pyruvate through 11 chemical reactions, that is converted to the glucose. Now let us talk about these 11 chemical reactions. Most of these, they are reversible. So starting from the first designated substrate, that is the pyruvate. As you know, the formula of pyruvate, that is something like this. Pyruvate is first converted to oxaloacetate. This conversion is irreversible, and for this conversion, a CO2 molecule is used, because pyruvate, that is having 3 carbons and oxaloacetate is having 4 carbons, so 1 carbon is added in the form of the CO2. Similarly, energy is consumed, 1 ATP is converted to 1 ADP, and the enzyme which catalyzes this first step, that is the pyruvate carboxylase. Because carbon dioxide is added to the pyruvate, so this process is called as carboxylation, and this step or this reaction is catalyzed by the pyruvate carboxylase. So the first product will be oxaloacetate. Then this oxaloacetate is converted to the second molecule, which is the PEP, phosphoenol pyruvate. And my dear students, again this reaction will be irreversible. And now this oxaloacetate, which has 4 carbon atoms, is converted to PEP, which is having 3 carbon atoms. It means the one CO2 molecule will be removed. Again, my dear students, energy will be used and an ATP will be converted to ADP. And this is catalyzed by the enzyme which is called as PEP CK, which stands for phosphoenol pyruvate carboxykinase. So that is the enzyme which catalyzes the second step. So the first step is carboxylation of pyruvate in the irreversible way. The second step, oxaloacetate, is converted to PEP by the decarboxylation, and that is also irreversible. Now, my dear students, in the third step, which is a reversible one, now this phosphoenol pyruvate, that is converted to 2 phosphoglycerate. And my dear students, in this process, water is added, and this is basically catalyzed by another enzyme that is called as the enolase enzyme. Now, my dear students, this 2 phosphoglycerate, then that is converted to one of its isomers, again through a reversible process, and that is the 3 phosphoglycerate. Now, my dear students, this is again catalyzed by an enzyme that is called as the phosphoglycerate mutase, and it is again a reversible process. Now, my dear students, this 3-phosphoglycerate is again phosphorylated, and that is converted to the next one, which is 1,3 biphospho glycerate or 1,3 BPG. Now, my dear students, this step again uses energy in the form of ATP, and it is catalyzed by an enzyme which is called as phosphoglycerate kinase. And again, ATP is converted to ADP, and the phosphorylation of the 3-phosphoglycerate that takes place. Now, this 3 biphosphoglyscerate, it is again changed to the next molecule, that is the glyceraldehyde 3 phosphate or G3P. It is again reversible in nature, and in this case again an enzyme is used which is called as G3P dehydrogenase. And my dear students, now this G3P is in equilibrium with one of its isomer you have already studied in the glycolysis, and that is dihydroxyacetone phosphate. And here this is catalyzed by an enzyme triose phosphate mutase. But you already know that dihydroxyacetone phosphate, no doubt, it is a ketoisomer of the glyceraldehyde 3 phosphate. But majorly the process of glycolysis as well as the gluconeogenesis that is taking place or that takes place with the help of G3P. So, my dear students, in the next step, the G3P is converted to fructose 1,6 biphosphate. This is fructose 1,6 bisphosphate. And my dear students, now this reaction is catalyzed by an enzyme that is aldolase, and fructose 1,6 biphosphate is generated. How many steps we have studied? This was the first, now this one is second, third, fourth, fifth, sixth, seventh, and eighth. The first and second steps they were irreversible, and all other steps they are the reversible steps. Now, my dear students, the ninth step in which fructose 1,6 biphosphate is converted to fructose 6 phosphate. And my dear students, this reaction will be irreversible, that is the ninth step, and it will be catalyzed by an enzyme fructose 1,6 biphosphatase. Phosphatase is the enzyme which breaks the phosphate linkages, and from this step, an inorganic phosphate is removed from the reaction. And my dear students, this glucose 6 phosphate is then changed to its isomer, which is the glucose 6 phosphate. So, fructose 6 phosphate is converted to glucose 6 phosphate, and this reaction is again catalyzed by an enzyme that is the phosphoglucoisomerase. And my dear students, there will be now the last step of this gluconeogenesis, in which the glucose is synthesized from the glucose 6 phosphate. So this one is the glucose. And my dear students, this reaction is catalyzed by glucose 6 phosphatase, and again one inorganic phosphate that is removed from the reaction mixer. Now it was the eighth step. This one is the ninth, 10th, and 11th step. Out of these 11 steps, my dear students, the step number 1, 2, 9, and 11. These steps are the irreversible one, and all other steps they are basically the reversible steps. Similarly, in the sixth step here, 1NADH is converted to NAD positive. Mean the reduced nicotinamide adenine dinucleotide is changed to nicotinamide adenine dinucleotide. My dear students, this process of the gluconeogenesis, it looks like the reversible process of the glycolysis. It is somewhat reversible, but it is not the exact reverse process of the glycolysis. There are some steps which are different from each other. And in this complete gluconeogenesis process, how many ATPs are used? One, two, and my dear students, this NADH, it is again energy rich, that is used and this one is the third ATP. So one, two, three ATPs are used and one NADH is used. You know, one NADH produces three ATPs. So it means in this complete process, total six ATPs are used. Three ATPs are directly used, and one NADH is equal to three ATPs. So total six ATPs are used. If you can recall, then in the glycolysis, only two ATPs are gained. The net gain of the ATPs is two in case of the glycolysis. So glycolysis produces two ATPs and gluconeogenesis consumes 6 ATPs. It means that the gluconeogenesis consumes 3 times more ATPs as compared to the ATPs which are produced in the glycolysis. So it is an energy consuming process already due to the prolonged fasting, starvation, very intensive exercise, and my dear students, due to inadequate intake of the carbohydrates, the body is feeling weak, the body is feeling less energetic, and this process of the gluconeogenesis is again consuming the energy in the form of the ATP. Then why this process happens if it will make the weak more weak, or it will make the weak into the weaker? Because this process is very, very important to maintain the proper blood glucose level in the body. The brain cells, the red blood cells, the medulla cells, and the testes, these are the cells that only depend on the glucose for their energy. So if you will not maintain the proper blood glucose level in the body, then these organs, they can be damaged. So that is why the gluconeogenesis takes place. Even though it consumes energy, it maintains the blood glucose level, which is very important. So that was all about the gluconeogenesis. I hope so this lecture will be very helpful for you people. In case you find any problem, you can ask in the comment section. Till then, take care.